think this is number 8. DNA replication. So when DNA replicates itself to make a new strand of DNA, or 2 new strands of DNA. How this works is an enzyme called helicase, right here, goes and breaks all the bonds between the double helix structure, so we have 2 old strands of DNA, like this. These are called replication forks. So these replication forks each have half of the acids or DNA pieces needed, and it sort of acts as a template. So what's next is, well, it's leading and lagging strand because it runs 5 to 3, and this one runs 3 to 5. We'll get to that in a second. So what happens is primase comes and lays down an RNA primer. Now the RNA primer is a piece of RNA that just connects as a temporary thing. DNA polymerase comes and bonds to this primer and runs in the 5 to 3 bond continuously, laying down new DNA, and then lays down the new DNA, yeah. Now in the lagging strand, it can't run that way continuously. It runs this way continuously, laying down one piece of DNA at a time. It can't run continuously this way because it is the opposite prime. So what happens is we still get the primer to lay down one piece of DNA, and then the DNA polymerase has to come just do a little strand. This little strand is called an Okazaki fragment. So what happens next is it lays down an Okazaki fragment and then goes forward and lays down another. This is quickly filled in by new DNA or RNA. Eventually, all the RNA is taken out or replaced by DNA. Then we get another enzyme to come and bind it all together, so that there is no RNA and all the DNA is bound together. What happens next is we have 2 new pieces of DNA called semi-conservative. And they are called this because they have an old strand of DNA and a brand new strand of DNA. It's good to have semi-conservative because you have less of a chance of getting mutations because you have a constant template backbone to go based off of to make a new one. That's DNA replication by Spin Levine.